My name is Hilary Papworth and I lead the ornamental crop research here at NIAB, and I am based at our glasshouse facility in Cambridge. Along with my colleague Ben T, I am leading research work funded by the AHDB in nutrient management in bedding plants. The aim of this work is to provide recommendations to growers for their crops and advice on best practice in crop production and monitoring. One area of work in this project is nutritional issues in primulas, specifically the cause and prevention of the physiological disorder leaf edge scorch. This crop can display marginal leaf necrosis, which is similar in appearance to scorch that you might see in high temperatures and light intensity conditions. The picture here shows scorch on the right caused by high temperature, and on the left, leaf edge scorch caused by nutritional issues. You can see the difference in that the nutritional problems affect the young leaves and very localized necrosis of the margin. Necrosis in foliage can be caused by deficiency of either boron or calcium. These have poor availability to the plants at different pH levels. Boron at pH above 6 and calcium at pH below 6. Our trial work looked at pH and availability of these elements to Primula to see if symptoms were caused. This was achieved by growing plants to flowering stage in different pH growing media, and at intervals sampling the growing media and the leaf tissue to see the result. Leaf tissue analysis confirms that pH affected the levels of both elements in the leaf tissue as we expected. Calcium and boron are what are known as immobile nutrients. That is, they cannot move within the plant independently. Both of these elements move with the flow of the water, from the growing media into the root and through the plant. Any factor which affects the movement of water will affect the flow of these nutrients to the part of the plant where they are needed. Transpiration is the most crucial factor in the movement of water within a plant. This is defined as the evaporation of water from the leaf cell and the loss of this water vapour out of the leaf stomata. In this video, I'm explaining some of the background to calcium movement in plants that was the background and basis of our trials.
So calcium nutrition is very well understood in a lot of crops where this tip burn or leaf edge scorch is a common problem. So brassicas, lettuces for example, these types of crops where the head is very enclosed, where leaf edges can easily burn because there is very little air movement, very little transpiration taking place in a closed environment. Primulas obviously are a very different crop. They don't have that kind of problem, but the same environmental conditions are going to be having an impact on the movement of calcium ions within the plant.
So because movement of both calcium and boron is affected by transpiration, we looked at growing plants in different humidity environments and with different water supplies. The results for plants grown in high pH growing media indicate that boron had no role to play in the cause of leaf edge scorch. However, calcium was indicated as a cause of the problem. Analysis of the leaf tissue showed that calcium was present in lower levels in the leaf where plants were grown in high humidity conditions, and this is where we saw the leaf edge scorch symptoms. So even if there was enough calcium available from the growing media or the irrigation water, high humidity can prevent the movement of this element to such an extent that it will cause damage to the plants.
Calcium plays a key role in the plant cell wall. This is from the tight binding of calcium ions to the pectins present in the cell wall. Calcium ions are involved in the stabilization of cell wall structures, which enable them to be rigid. They also control the activities of wall enzymes. Without sufficient calcium, the enzyme polygalacturonase will break down pectins, causing cells to die. Another function of calcium is to aid the transport of other ions across the cell wall membrane. However, it is its properties as a structural component that are important in leaf edge scorch.
We also found that the same symptoms were seen where the water supply to the roots is poor. In our trials, we looked at limiting the water supply to plants to look at the effect of root pressure on the movement of calcium in the plant. Root pressure is the force that helps drive fluid upwards in the plant in the water-conducting vessels. It is primarily generated by osmotic pressure in the cells of the root. The force of root pressure to drive water is not as great as that caused by transpiration, but it is important in small plants, particularly when the plants are transpiring less, for example at night or in the winter. If water supply to the roots is interrupted on a regular basis, allowing the plants to wilt, root pressure will drop, affecting the flow of water and subsequently the movement of calcium in the plant. As root pressure is mainly important when transpiration is normally low, having lower humidity in the glasshouse will not help the movement of calcium, so will not increase the levels in the leaf. In our experimental work, we demonstrated this by limiting the water in some treatments from midday onwards. This meant that some plants had low root pressure going into the evening. These plants had the lowest calcium levels in the leaf overall. So we found that it is the combination of a moderate but consistent supply of water to the roots and lowering of the crop height humidity to below 80% that is most beneficial for the calcium supply. Under these conditions, leaf calcium can be increased by between 20 and 40% with no additional fertiliser requirements. Monitoring of humidity must be at crop canopy level. Overall glasshouse humidity and air movement can be good, but it is at the plant level where the change is needed. We achieved this by increasing the spacing between plants.
On the left-hand side are the plants that were spaced out 10 centimetres between pot centres, and the plants on the right-hand side are from the very closely spaced trial plots. Now you can see there's obviously a very big difference in the quality between the plants. The nicely spaced plants are much better shape, they've got much better formation of the leaves. They've made generally just a nicer, more saleable plant. The plants on the right-hand side where they were closely spaced, we've got much more uneven growth. There's been a lot of competition between the plants for light, and it's caused a lot of stretching on the leaves, and they're generally not attractive.
Humidity will also be affected by the growing surface, so we would expect an increase where capillary matting is used. In these situations, air movement and ventilation need to be considered, as increasing these will also reduce humidity. We should note that our observations on the size and overall fresh weight of the plants in the trial showed that reducing humidity in the crop produced a 10% smaller plant than those in higher humidity conditions. Leaf edge scorch is quite different from the symptoms of iron deficiency, which are also common and appear as distinctive yellowing of the young growth in Primula. We found this can be readily treated by applications of sequestered iron products. We found that 4 applications at weekly intervals of a liquid product containing 2% sequestered iron was sufficient to combat symptoms of this deficiency. So our key findings so far on our work in leaf edge scorch in Primula are that calcium deficiency is the most likely cause of these symptoms, that the pH of the growing media has a less significant impact on calcium deficiency than the environmental conditions, that reduction in crop height humidity has a positive impact during spring and early summer, but root pressure may be more important than reduced humidity during the winter. Full details of our experimental work can be found in the final report of the AHDB project POBOF 003. We would like to thank the AHDB for the funding for this project and our industrial collaborators ICL and OMEX.